Cool. I'm good. So let's jump into this one. If you're good, I'm good.
I'm not good.
Okay.
I can add them on though, and you do the intro. I'm like 2 monsters deep, man. I'm gonna add them on.
Sounds good.
All right, here is Dr. Jamie Craggs and Keri O'Neil, two of the people that are at the forefront of coral spawning. I will be right back.
This is so awesome to have both of you on here. It is still early.
You got a long way to go.
Exactly. I was like, man, 2 monsters in 3 hours. He's gonna be ruined.
If you'd have seen him on the FaceTime chat we had about 11:30, you'd have been like, "You aren't gonna make this 24 hours. There's no possible way."
Oh my god.
He was out. He sent me a picture of him with him and his girlfriend were out hanging out last night since they weren't going to get to see each other for basically 24 hours. And they had these drinks lined up. I'm like, dude, you're in trouble if you drink all that. And sure enough, you could see it in his eyes this morning when I was talking to him, but he'll be okay. He's young.
He's young. He can still do that.
Exactly.
What's the strategy? Are you tag teaming it? I mean, you got to go for pee breaks and you got to eat and...
Well, we got 7 o'clock comes around. That's going to be an hour break for Salem and I, where we can go get some real food and actually stretch and walk around. And we got Mark Levinson's coming on with Sanjay and Mike Paletta, and they're going to take over for an hour. We figured the three of them can easily talk for an hour about anything. So then at 5 a.m. we have the UK boys coming on with Ryland and Thomas are coming on to have a chat with Paul Hughes. So I know that they're not going to have any problems having a good chat for an hour. And of course, if I'm here for half of that or a quarter of that or whatever it is, we'll get involved with the conversation as well. But oh my gosh, like I said, Salem and I were talking about having you two on. This is like one of the episodes that I'm so excited to have on because I love what you guys have done. And I've learned so much from both of you. Keri more than Jamie, but I've talked to you just a little bit back and forth with messages. And thank you for any insight that you've given me. Yes. So just so everybody knows here, Keri O'Neil, she is the I'm going to let you say it. Tell us who you are and why people need to listen to what you have to say. And I want Jamie to do the exact same.
It's all right. It's a long title and it changes constantly. The Director and Senior Scientist of the Coral Conservation Program at the Florida Aquarium. So I manage our off-site coral conservation facility where we breed corals native to the state of Florida and then settle the larvae and raise the babies and put them back out onto the reef for restoration. And we'll get more into the nitty-gritty details of that and all the crazy things we've accomplished over the past, I was thinking it's been 7 years since I went to visit Jamie and kind of get the wisdom from him and bring that back to Florida and set up the first systems at our facility. So it's crazy.
And we are in the height of the Caribbean coral spawning season, right?
Yes, we are. I just got a text 20 minutes ago. No spawn today.
That's good and bad.
I would have been doing this live stream from the lab had it happened today.
That actually would have been really cool. And Jamie, tell us a little bit about you, buddy. I mean, you've been you pioneered this whole thing. Like Keri said, she learned a lot from you, and I think it's amazing the work that you've done. And just tell us a little bit about what started you off in this crazy ass journey of spawning corals.
Yeah, I mean. Thanks for having me on. It's great to be on with Keri and chat about our passion. We are proper coral spawn geeks. Life's got pretty busy. I've got 3 jobs now, which is pretty hectic. But the journey started at the Horniman Museum and Gardens. It's a small public aquarium, very small. I always say it's small, and people I've been to small aquariums. This is really small. We can sprint around it in 12 seconds. It's tiny. But we started on this journey back in 2012, trying to figure out how we could spawn broadcast corals, basically. And within 8 months, got our first spawning. Thought we're definitely onto something here, and it's just carried on growing from there. And I think it's insane the kind of level of work that can now happen as a result of land-based spawning, and there's so much potential. We're literally barely scratching the surface on it. And that's exciting, right? I mean, that's what it's all about. Yeah. And then, just 3 jobs now. So we set up Coral Sporting Lab. That's had a sort of a brand change to Coral Sporting International recently. Nice. And more recently, I've started working for Mars Sustainable Solutions. So one of the things about working in London is it's amazing doing all the sporting work, but then following what Keri's doing, she gets to take it full circle and literally plant those things in the ocean. And that's the whole point, right? So now working with Mars, I've got that kind of in situ restoration side. So it feels great. We can do a bit of research in the museum, support others through Coral Sporting International, and now actually engage in true restoration as well. So, but it means not a lot of time for sleeping.
Well, this is kind of a lifestyle, when you're in the aquatics, especially in the coral side of it. It's a lifestyle you have to embrace. I mean, if you want to do what we do, heck, Keri, when we first met, I think our first time meeting, we talked for 5-6 hours. I think Richard recorded it on Reefs.com at one point and we just had a session with her and I talking about all this stuff and just getting to know each other. What you've taught me is just, it's invaluable. It's amazing.
Every time I'm like, I'm going to go visit Chris and Amanda. I'm like, do I have 8 hours today? Or I'll have to tell somebody can you text me in 2 hours or else I'm going to for 6 hours. It's like when you go on a blind date and you need somebody to call you and check in.
Well, the thing that I loved about Keri, she taught us and she helped us get our apexes set up. She came to us, was like, I want to spawn acanthophilias. And of course, that was my wife's wheelhouse right there. It's her favorite of all time species of coral. And of course, Amanda jumped right onto it, and God bless her for being over there for a month and a half straight, watching these things, nothing happening. And then when it finally happened, it was at 6 o'clock at night, and we calling Keri, we got babies. And the next thing you know, she's on her way out helping us collect these things and learning about the craziness of acanthophilias, which I think are probably still your most difficult species to date. Is that right, Keri?
I'd say they're comparable with the Dendrogyra. The pillar coral, Atlantic pillar coral, I would say is more difficult than them.
Okay.
Just in the rearing, because they're tiny, but it's similar to the acanthophilia. They're just these tiny goo balls for so long, and they're so fragile, and they just don't grow very quickly, so you don't get out of the goo ball stage for months and months. They're difficult to raise. Yeah.
But collection of the actual spawn.
Yeah, because
you were saying it.
Broadcast eggs into the tank, and they're just loose eggs everywhere, and they're very fragile, so trying to filter them out is very damaging, and you're just sitting there trying to pipette them out or gently gravity feed them out, and it's a pain in the butt. There's definitely better ways to do it, but we didn't know about acanthophilia at all. We didn't know if a bundle was going to come out of their mouth, or whether they would be gonocores, male and female separate. We had no idea what was going to happen. And then come to find out they're hermaphroditic, male and female, so sperm and eggs in the same polyp. But it just all comes out loose, not.
Wow. That's amazing. I wouldn't have thought that. I would have thought there would have been a big bundle come out. Yeah. Wow.
Yeah, it's similar. So Musa angulosa in the Caribbean we found is like that as well. It's just sperm and eggs loose, and then the eggs are fairly tiny, and they're kind of neutral too. They don't float to the surface very well unless it is absolutely stagnant, not even a little bit of air across the top, and then they will eventually come to the surface, but they're a pain in the butt to get out of the tank.
The Catallophilia here. So a spawn Catallophilia a few years ago. I'm in the home system. We've got a little tank at the side, and they released this huge. It's can be the length of that size. It's huge, the biggest bundle I've ever seen, with thousands of eggs in a single bundle. But the second they break apart, same deal. They're almost neutrally buoyant. It's a nightmare. So you have to contain them at the spawning, otherwise you're, like you say, age is just.
And at least they have that chance with the bundle. The acanthophilia are like, no, you're going to sit here and pipette eggs for hours. And at least in my tank at home, it's a glass tank. So like if you're shining a light, you can see. Chris had them in a fiberglass tank. And Lord, you're like trying to go on the side and like so bad.
we ended up getting these little goggles that you put them on your head, and it had these big magnifying glasses that stuck way out, and we're like looking down in the tanks trying to get these things, and each individual egg, literally, you sat there, Keri, with us. I mean, we had my son doing it, my daughter doing it, Amanda, Keri, Samantha came over from Aquaholics, and we're sitting there with these pipetters individually sucking out these eggs. And I'm like, how we got so many of them was beyond me. I mean, we literally did it for 4 hours. I mean, at that point, they were already dividing. How many we destroy, in the process. But we still had a blast with it. We learned a lot. And I'm ready to do it again. This year, I'm ready to jump on it. But I just got to get the system designed properly this time because it was just something tiny. It needs to be done right.
Yeah. We had gotten the word from Don and Lou that they had seen mature eggs, and then I actually had taken some histology samples. So Chris sacrificed one. Well, we cut it into pieces. We cut two of your acanthos into quarters.
Four.
It was far more painful for you.
That was heartbreaking. But the thing is, we learned how to frag them.
Pie pieces, and we're like, okay, let's see if these pie pieces still live, and I'll take a pie piece to do histology. So I brought it home and I fixed it in formalin, and I dissolved the skeleton in acid, and then I just dissected it, and I was like, we gotta hurry, Chris. We better get ready now.
And what color are the eggs? Are they pink or are they more yellow?
Very, very light peach. Yeah.
Okay.
Yeah, peach, very peach.
But they had a distinct peach color when I dissected them. I was like, we've got to get on this now.
I mean, how cool is that? I mean, with homophilia, they're yellow. With trachophilia, they're green. It's just amazing. What the hell does all that mean?
No.
Yeah, I mean, what does that correlate to? Is it like protein concentrations or something?
We worked with Dipsastria in Qatar earlier this, was it last, losing track, I think it was last year. And what's crazy is they release a whole rainbow of colors. And it's not like, and you get purple, pink, green, yellow, beige.
That's cool.
But you think, is it coming like purple from one colony, green from another? It's not that at all. So the same colony releases these multicolored eggs. It's beautiful.
It's like Easter.
It's like a nutritional thing. Like maybe if there's a deficit or an excess, or like they'll produce different colors in the egg. That's a weird one, huh?
There's a study in that for sure. What does it all mean?
Yet another study.
Yeah.
in the comments, somebody asked about the pieces surviving, and they did. I still have 2 of them in my tank, and the crazy thing is, even the pieces spawned again this year.
Yes.
So it was like the quarter pie piece that was the first one to spawn, and then it spawned again this year. I was like, the pie pieces live, and it's very funky looking, but it's formed a circle again now, and one side of it's really big and the other side's really small, but it did live and it spawned, so it's doing it.
So do you get like net more spawn from having more pieces, or does it correlate to the size of the polyp with solitary ones?
So my thought would be, so the eggs and sperm develop in the mesenteries, around the polyp. So if you cut it in quarters, you only have a quarter of the number of mesenteries. So I would think that you quarter
the
your
same amount, basically, if you have all 4. Yeah. Interesting.
Over time, you get more as it grows. But yeah,
sure. Yeah.
It's wild, absolutely wild. But I was actually helping somebody with acanthophilia spawning, and they didn't know exactly what to come, and I helped them go through the process and told them, okay, you got possibility after November full moon, after December full moon, and then I honestly believe 100% that after the January full moon is when the really big event happens, because I haven't broadcast this to everybody, but it might as well now. It's been a year since it happened, more than that. We had the biggest spawn in January, after January full moon, and we weren't ready for it. We didn't even think about it, and my dumbass put 100 acanthophilias in 150 gallons of water, and these damn things were spawning, and the whole tank turned milky white. You couldn't even see in the aquarium, and it's just like, oh, I can't believe we didn't come over in the morning, because if we did, we probably would have saved everything. But we ended up having a complete catastrophe. It was a lesson learned.
Yeah.
But it actually was really cool to hear from these people that did it, that the exact scenario panned out. They had small spawn in November, after November full moon, a bigger spawn in just after December, and then a massive spawn. And the thing that was really cool to hear was that they settled differently each time. Like, we had 2 weeks before they settled, right, Keri? Yours were 2 weeks, mine were 2 weeks.
Yeah, they were just slow to finally put their butts down, yeah.
Well, after the January full moon, that was the same scenario with the people that I talked to. After the January full moon, it was like they didn't even care. They had no preference for where they were settling, and within 5 days, they were all settled, and they had these collages of tiles that were just completely covered with baby acanthophilias. And I'm like, oh, cool. So we need to prepare for January full moon from now on and make sure that we are really ready for a huge spawn come January full moon. So that's just data that nobody ever knew about. And I wouldn't be doing it if it wasn't for you and Jamie. I mean, you guys are the pioneers. You guys are the king and queen of coral spawning. And I'm just grateful that I got to know you guys and we were able to help us and feed the passion that I have for it.
Well, it's been fun, working with you and Amanda and just watching your passion for it too, and just being able to crack the code on acanthophilia. And you guys get such beautiful, beautiful corals. So going over there and being able to be like, you should do that one, that one. And you guys were very generous to give me some for my home tank as well, and going in it together and going on.
We were happy to.
Together has been fun.
And it is so true that in America, you guys get all the good stuff. In the UK, we can't get any. I mean, it's illegal to bring acanthophilia or trackies from so many different places around the world now. And you see it's like eye candy, but it's like, oh, you can't have any of it. It's the worst.
So
I wanted to ask Jamie.
Go ahead. Yeah, you do yours. You do yours.
I was going to say, I wanted to ask Jamie, what is new and up and coming for you? What are you working on that is something you haven't really talked about, if you're willing to talk about it? Because
there's one project that Keri knows about. I can't talk about it to give all the details, but we've been working on a very, very rare. We think probably the rarest coral in the world. And trying to start a biobank with that, and we cracked the code to spawning that about 18 months ago.
Wow. And
we're building the population back up from that. So at the moment, the population is 121 individuals that we know of. So yeah, that's been quite a lot of work. So that's back in London. We've been working on that. And then we've been focusing a lot on temperate species now, trying to use some of that knowledge of spawning, and a lot of it is inspired by Keri's work and her team. The fact that you work with Florida Reef Track, you spawn them, get a chance to put it out. We were like, we live in London. We're not going to go and fly to other areas of the world, so let's start focusing on British, UK species. So we've been working on a Gorgonian that you get in the UK. And we managed to spawn that last year. We just got government permits to plant those out in the ocean, which is great. So I've got a PhD student working on that. She's going out in 2 days, actually, to plant the first babies out. So from the Horniman, that's kept us busy. But we've also been looking at, actually, Chris, I don't think we've ever met, Chris, by the way. I don't think we have or not. But I feel like I've known you for years because I've watched so many of your videos. And we've obviously had messages. But the paper that came out a few years ago looking at Cropper tenuis and how it's now tenuis in Fiji and Kentai in Australia and Bifera in Indonesia. We thought, well, why don't we look at that from a reproductive side of things? So we've started creating outcross lines, taking a sperm from tenuis from Fiji and fertilizing it with Kentai. We now know that Bifera actually, so the the tenuous and Kentai spawn in November, December. Bifera actually spawns in March, April. So we've not managed to cross those yet, but that's going to be the goal. We've made a tenuous Kentai tenuous cross. And the goal with that, we want to get those F1s 3 years old. If they're fecund, are they viable? And then it flips that whole question on its head. What is a species? If a species can be viable and create F2, genetically, they're similar or they're distant apart. We'll call those separate species. But reproductively, if you pull that back in and say, well, what is a species? So we thought that'd be a bit of fun. We've been working on that over the last year.
It's very interesting. So the Gorgonian species that you spawned, is that photosynthetic or is it non-photosynthetic?
No, it's a non-photosynthetic species. It's gonagoristic. Well, it's been said that it's gonagoristic, but we had sperm and eggs come out of a single individual last year. So we need to do a bit more work on that. But yeah, Unicella vericosa is the species. And we made a kind of a tweak on the system. So we have alternating tidal patterns. And so we sort of scale up the flow and then we drop it back down and scale it back again. And there's some really nice work done in the Caribbean in the sort of 70s, 80s, looking at flow velocity. And ultimately the flow kind of bends the polyp and that drives feeding efficiency. So there's this real sweet spot in centimeters per second, that if it's too slow, basically no food's going in. If it's too fast, the food whips out, but there's just this absolute sweet spot. And the pinnules on the tentacles, they're basically slowing that flow velocity down and driving feeding efficiency. So with that in mind, we designed the system to think about knowing like 10 to 18 centimeters per second is that optimal sweet spot for Caribbean species. So we designed the system to replicate that. And then we have an automatic dosing system that puts the food in at the optimal flow velocity.
That's cool.
So we've been following the kind of growth rate. In the wild, 1 centimeter a year is the growth rate. We hit 3.5 centimeters in the first year. So we're definitely on the right track with that, I would say.
There was a reason why I asked you that, and it's going to stem into what Keri and I have discussed, and I'm sure you've had this discussion with others. If it was a photosynthetic species and you were planning to get back into the wild and you were growing it under LEDs, what do you think the percentage of survival rate is going to be without getting true natural lighting that the environment provides versus what we think is good enough for these animals to grow, which we obviously know works? But what's going to happen going back into the wild with these species? And I know Keri knows where I'm going with this, and I want to hear from both of you. What do you think needs to be done to combat the issues with outplanting corals that were grown under LEDs?
Keri, do you want to pick it up, or I can give my...
Go ahead. I can talk for an hour just on this topic, so maybe you should get some words in.
So my reference point to this is work that I was lucky to go out to Palau for a couple of years with one of my PhD supervisors, James Guest, who runs Coral Assist Lab, and he's doing some amazing stuff with his lab. Looking at this question about human-assisted evolution. Can we selectively breed thermally tolerant individuals? I think they've shown for the first time that it's a multi-generational inheritance. So they, in 2018, I was part of the team to ultimately create high cross, high low, low high, low low. These genotypes were thermally stressed to rank them. And the high crosses, when you take eggs from high individuals, sperm from high individuals, they create the strongest individual F1s. What they then follow through is that inheritance passes multi-generational. So the F2 retains that thermal resilience, which is, it's such an important part of this puzzle about where can we go with this. James is always pains to state it's a marginal increase on thermal resilience. This isn't the panacea. It's not going to save the world's reef. It's a single species, but it gives a little inkling of where this work can go. the reason why I'm talking about that there is obviously we did that in land-based systems. A paper came out looking at if you keep them in land-based system for 5 months, then plant them out, 11 months, plant them out, and then 13 months, plant them out. Look at the survival of the juveniles at each stage. What was interesting is each stage, that the photo stress is really high. They can survive, but they definitely bleach, so that photo acclimation is absolutely critical. And that was grown under sort of a standard, I'm not going to say the brand because that'd be mean to the brand. But it didn't, at the end of the day, LEDs aren't going to create the power of the sun, and that's fundamental. There's no physics that's going to put that energy through an LED. So having mechanisms to kind of manage that transition. And Keri, you've got way more experience than me with this, but I know that you've experienced some of that as well. Well, a lot of that. So I hand over to you.
So just a little bit of background. Before I came to the Florida Aquarium, I worked at the National Aquarium in Baltimore, and all we did, I started there in 1999. So all we did was grow corals under metal halides. But I wasn't putting corals back out on the reef. And then when we started this work at the Florida Aquarium, the brood stock or colonies or parents were kept under LEDs because that's how we do the spawning systems. But all of the offspring were settled and raised in greenhouses. Now, mind you, these greenhouses have a twin wall polycarbonate roof. I mean, they're Cat 4 hurricane rated greenhouses. They have a very solid structure and twin wall polycarbonate. And that does block out actually a bit of the blue end of the spectrum. So it's a bit of a red shifted light, but it's pretty full with just a little less blue. But no UV because the UV doesn't penetrate the polycarbonate. And we've put out, I don't know, upwards of, either we or have given to partners, have planted upwards of 25, maybe 30,000 offspring at this point from our facility. Only one instance did we have severe bleaching after outplanting, and that was early on, and they were planted in less than 12 feet of water. They were planted at 8 feet, which is very, very intense light. And UV is still present at that depth. UV is actually doesn't penetrate water very well. It gets down some, but UVA gets down, B doesn't do as well, C definitely doesn't. So when you're planting corals in very shallow water, it is critical to have some sort of acclimation phase. Now, whether that's just overall intensity of light or actually a UV acclimation is something we've kind of been trying to figure out. The majority of coral outplanting in Florida tends to happen at 15 feet plus, 20 feet, 30 feet. So we tried to, and we've actually had no issues with corals going directly out from our facility to 20 feet of water, no bleaching. They might get eaten by parrotfish or have other problems, but they don't bleach. So that being said, then we recently expanded our facility. We have a new 4,500 square foot facility that is all LED lighting. So now we are also settling and raising offspring completely under LED lighting. And the reason I kind of made that operational shift is greenhouses are great for some things, but I can't control anything about the spectrum. I can control the intensity by adding shade cloth, but I cannot change that spectrum at all. And it is not at all a spectrum that you would think a coral would want in any way, shape, or form. They still grow. They do fine. I do think that algae is probably a bit worse out there. But they're also very, very hot. Keeping a greenhouse cool and then cooling the water inside of the greenhouse is actually very energetically costly, and it's turning out to be a bit more energy efficient to just have an air conditioned building with the tank with LED lighting. So we now have many systems where we are settling the corals under LED lighting and raising them under LEDs completely. And that has still gone fine. There's still, if anything, we can tweak that intensity even more. We can tweak that spectrum and drive even a little bit of a faster calcification rate, I think, from qualitative observation. And recently we have outplanted now corals directly from that building. Same thing. We actually outplanted corals right before the 2023 marine heat wave. And we were doing a project where we were trying to get the shallow water bleaching. So we had done, this is a project done by Rachel Morgan, who previously Rachel Seraphin, but she's married now. So Rachel Morgan, and she is our coral propagation manager now. And she's been with me now for almost the whole time, about 7 or 8 years. And she's now working on her master's. And we were looking exactly at this. Like if we did a UV acclimation phase before outplanting. So, the corals protect themselves from UV by producing compounds called mycosporin-like amino acids, or MAAs. And they can, if you expose them to UV, they can kind of switch that on relatively quickly. Like you can start detecting MAAs in their tissues within a matter of a few days to a week. So we were like, all right, let's just do a month, of a slow ramp up of UV exposure. And then let's outplant them to shallow and deeper sites.
Awesome.
Sounded like an awesome project.
So I've got a little bit of a different question about, resilience and adaptation strategies. So, like I was in the Bahamas last summer at Cat Island, and they had an outplanting facility there where they were doing like trees like Palmata and Cervicornis. I think it was like a shed aquarium, and they had described they had a ton of losses there. I don't know statistically what that was, but UV is one thing when it comes to adaptation from an ex situ to an in situ environment. But I think about like the different symbiont associations too, whether it be the symbiodinium or bacterial populations. And obviously corals are very plastic and can uptake things from the environment and switch them out as they see. I think, at least from what we've seen so far, a lot of these ex situ systems have really different microbiomes, and they don't have all these, like the free-floating diversity of symbiodinium like you would get in the ocean. So, would it not benefit to maybe have some type of like buffer period to where you take spawns in a captive system, put them in like a flow-through system, like on like a beach type of area, or like an area where there would be water coming in? I don't know. Like, I feel like that could potentially be a lot of the die-off too, as dysbiosis occurs potentially. I don't know.
So, I just want to clarify, because there's not die-off. Sometimes they get eaten by parrotfish. Like, we generally in the first year have 90% or better survival.
Okay.
And if they're big enough.
Yeah. What age do you plant out at that?
We try to get them to at least 2 centimeters in diameter.
Okay.
Which for us, depending on the species, is about a year. Mm-hmm. And we generally have very, very good survival until some other major event happens, like a marine heat wave. I was trying to get to before Salem. Sorry.
I did not mean to interrupt you. Sorry. Yeah.
At the different depths, none of them bleached. Even the controls that had no UV. So we didn't. Sorry, Rachel, I'm spoiling the study. And then the 2023 heat wave came, and they still didn't bleach. So they actually did great, and never bleached, and lived through the 92-degree water temperatures on the reef, and we're fine. And they're actually still doing really well. So we actually have outplanted a lot directly from land-based facilities without all these extra steps. Like people do, yes, if we needed to add the extra steps, definitely. But if you don't have to add the extra steps, that just makes everything more expensive. So everything we do now is trying to keep it as cheap as possible to grow a 2-centimeter coral and get it back out onto the reef. So adding another whole set of facilities that can handle 1 million corals a year that have to go through flow through for 3 months before they go out to the reef, it's just, it makes it even more complex. So if they can go from land straight to the reef, that's ideal. We've also done periods where we acclimate them on a coral tree for a month, 3 months, whatever, before the reef. Doesn't really seem to change anything. We thought that might change the predation. We thought they smelled good because they were grown on land or something, and it didn't really change it. So many things. So sorry, Salem, the microbiome. So
it's okay. Sorry.
In our tanks, we seed our tanks with live rock from the Florida Keys. And we also keep parent corals in our sumps. Our sumps are bumped out to the side, so they also get light. So they're, in theory, providing the microbiome to the recruits that we're rearing in that tray. And we've done a lot of work with students at the University of Miami, looking at the symbionts in our juvenile corals, as well as taking microbiome samples of the tanks to see what symbionts are available. So all of that work is still being worked up by a PhD student at the University of Miami, but we're looking at trying to see if we can make the corals take up durstinium, which gives them a little bit more thermal tolerance, but then also keep it, because we've found that they primarily will take up durstinium early, but then at about 1 year of age, they start to switch. And that is even if they're in the same tank, and looking at the microbiome of the tank, those multiple symbionts could be available. But sometimes, like for our acroporids, they're taking up Symbiodinium fiti from day one, and they're keeping it. So there's still so much that we don't understand about the microbiome in the tank, what ends up in the baby coral. And we just finished a study with Moat, UNCW, Nova Southeastern, and the Florida Aquarium. We all took the same larvae batch, settled them, raised them under 3 different light treatments. So we had natural light if the facility had it available, and a typical AB plus type spectrum on a LED, and a fuller spectrum on a LED. And I don't want to spoil the results, but and we looked at algae cover as well as survival and growth rate across 4 different facilities, all running the same light programs, same. That's awesome. So all these different we did our own sort of method of husbandry. And the results were surprisingly similar across the different facilities. And, you can totally grow baby corals under LED lights, and they do just fine. But we also took microbiome samples of those corals at multiple time points. So that is not processed yet. But I tell you, from what I've seen, and we've taken microbiome samples for various things. We do a lot of antibiotic treatments. We've done before and after antibiotic treatments, yada, yada, yada. It's so diverse, and it changes so rapidly.
So quickly.
Like, you could test on Monday, and it's going to be completely different from what it is on Thursday. You can't just take one sample and go, that's the microbiome of my tank. It's this constantly shifting thing. And if I could sample 10 pillar corals, they're all going to be different for the most part. They might have some similarities. And I could treat them all with an antibiotic, sample them again, and they're all just different again. Like, so we don't necessarily know, like, to the detail of what exactly is good or bad. Do we know that different is bad? Interestingly, the pillar coral in our tanks were compared to wild pillar coral samples, and they're like 90-something% similar. It was statistically different, right? But I was like, but it's 90% similar. And
Exactly.
That's cool.
It is very cool.
So we start to look at that kind of stuff, but that's like in its infancy. But we are really interested in how these recruits are developing their microbiome. How is that comparing? But here's the thing: as soon as I put it back in the ocean, it's just going to completely shift again.
Yeah.
And we've done some work on that too, of like test it before and after, and all of these things are currently like being worked up by PhD students, mostly at the University of Miami. So hopefully we'll know more about that in the next year or two.
I would add a few things to this as well. So before doing spawning work, we were focusing at the museum with at the time Newcastle University, looking at disease states, and we published a paper. It never really got that much traction, but I really liked the paper. It was looking at ultimately shifting from a stress state. So it wasn't necessarily disease. It was just in a tank that was really badly looked after. And, we moved them in, we looked at 3 species, I think, and then tracked the microbiome shift over 4 months of those 3 species. And it's amazing how transient it is and how rapidly going from a stress state into a healthy state, as long as you remove that sort of external stress, whatever that might be, the coral adapts and shifts back very quickly. So that's one thing, a comment on the microbiome. We have got 2 studies which are nearly there to write these up. One was we created intergeographical hybrids between Singapore and Australia. So we used Hyacinthus from Singapore, then we use Hyacinthus and Meliopora from Australia, and we basically phase shifted the spawning of Singapore, which normally goes in March and April. We moved that to overlay with Australia in November, December, and the idea being that Singapore is much warmer than Australia. You hit sort of 32 degrees in the summer. Australian corals, 29.5, they're maxed out. And so we wanted to use eggs from Singapore, sperm from Australia, and vice versa, create both interspecific and intraspecific hybrids between the different. So we made Hyacinthus Meliopora hybrid crosses as well. And then we took the adults and we took the juveniles and we put them through a thermal stress experiment. We killed them about 25 days in, and looked at the metabolomic profiles of both the adults and the juveniles. One, we found that the juveniles are so plastic, and I think this is a really important part about restoration, is we have to intervene when they're young. Give them that ramping up and stress, because they seem to be able to adapt to that stress, and there's some evidence to show that once they've adapted, they can draw back on that memory later on in life. So there was a study from Thailand from about 10 years ago. The second study that we're nearly there, again, publishing, was looking at, rather than a genetic component to thermal resilience, what is the epigenetic component that goes on? So we created a pool of genotypes, so we weren't looking at crossing eggs from one individual sperm for another. We just mixed genotypes for, I think, 5 or 6 Meliopora from Australia again, settled them, and then from the day they were settled, they got separated out into 3 feed treatments and a control treatment that never got fed. We grew those out for 18 months, and then we put them through a thermal stress experiment. And what we found is, if you feed them and certain foods, it's not all foods, we put a commercial diet in there. Again, I'm not going to say who it is, but that was that bleached and died quicker than the control of not feeding at all. But when we started using live phytoplankton, live zooplanktons, we uplifted their bleaching tolerance. So this is a driving factor specifically related to the environment, not the underlying genetics. So it is ultimately ridiculously complex. That's the take-home message. You can't just go, it's one thing or the other. Everything plays a role within this.
I get goosebumps, like listening to you guys talk. It's like, oh, this is what I love. I'm sitting here going like this, going, wow, it takes a lot to give me goosebumps. And I've been listening to you guys, and it's just been spot on, exactly what I was hoping to get out of this conversation. You guys are awesome. Love the information.
So I think, Jamie, you've mentioned hybrids a couple times, and Keri just had a pretty groundbreaking involved with hybrids. Why don't you talk about that, Keri?
Well, there's been two kind of hybrid things that we've been able to accomplish. The first was we were able to cross the Atlantic staghorn with the Atlantic elkhorn, so an actual hybrid between two species. And although that hybrid is wild occurring, there's only two known true hybrid. It's called Acropora prolifera is what people refer to it as. Two of those ever actually documented in the state of Florida, but some other islands in the Caribbean, you can find a ton of hybrids. So going on the principle of hybrid vigor, where you hope that the hybrid offspring might have more resilience than either of the kind of purebred parent species, we created upward of 700 different hybrid genets, using either the staghorn as the egg and elkhorn as the sperm, or elkhorn egg with staghorn sperm. We were able to do that in both directions, which previously people thought it only worked in one direction, and turns out that's not true at all. They work perfectly fine in both directions. Settled those and grew about 700 of them. And again, working with the University of Miami, had a student testing the thermal tolerance of the hybrid offspring versus purebred elkhorn coral and staghorn coral offspring. Turned out that they didn't show much heat tolerance in an actual sea bass. Is a way that researchers test the thermal tolerance of corals. It has its positives and negatives, let's just say. So they didn't actually test out that much better than regular elkhorn coral in sea bass. However, we did get permission to outplant them. At first, there wasn't going to be permission to outplant them, but Andrew Baker got a permit to put some of them on an artificial reef structure off the coast of Miami. And they went through last summer upwards of 14, maybe even 18 degree heating weeks. So we measure, the way we measure thermal stress on a coral is in degree heating weeks. So the number of degrees and the number of weeks it's been above its normal average temperature. So in the summer of 2023, the reef temperatures in the state of Florida actually broke the scale on degree heating weeks. I think the scale only went up to 14 or something, and we got 18. I apologize if I got the numbers wrong. Yeah, it was really hot for a really long time. So the moral of the story, it was literally 92 degrees on the forereef. And that killed the majority of elkhorn coral that was still alive in the state of Florida, except for those in like the very northern Florida Keys and Miami and Fort Lauderdale area. Everything farther south, gone. So we were hoping to make this hybrid. Long story short, they were outplanted in Miami and experienced a high number of degree heating weeks last summer because they're in an inshore, really hot area. So we put them there on purpose. It's going to test their resilience, and they didn't bleach, and they look beautiful, and they're still growing, and they're amazing. So still a lot. And now, people are starting to think like maybe we should put some more out somewhere else and see how that goes. So that was our first hybrid. More recently, also with the University of Miami, we do a lot with the University of Miami. We are like the husbandry experts, right? They are the research experts. So we work well together. And they imported, so Andrew Baker and his Coral Reef Futures Lab imported, I think it was 13 fragments of elkhorn coral, Acropora palmata, from Tela Bay in Honduras in partnership with Tela Marine. And so living fragments, and these elkhorn coral thickets, they're actual like thickets of elkhorn coral in Tela Bay in Honduras, are stunning. And they're near shore mainland, get a lot of runoff, get very, very hot during the summer just because of the way the water flow current patterns are in that area. That bay gets very hot, and these elkhorn coral are thriving. So, why exactly, we don't know yet, but hoping that maybe they have some genetic link, or maybe their symbionts are just super strong. Andrew imported 13 of those, and 7 of them came to the Florida Aquarium literally the day they landed, drove them straight to our facility. They spawned last July at our facility on the same nights that we had Florida elkhorn coral spawning.
Nice.
So we were able to cross Honduran elkhorn corals with Florida elkhorn corals.
Nice.
Now, if you look at the genetic data of the elkhorn coral population, it's kind of divided into 2 provinces. So there's the Western Caribbean and the Eastern Caribbean. And Florida's population is connected with the Western Caribbean. So the whole coast of Central America. So you have, Belize, Mexico, Honduras, that whole Mesoamerican barrier reef seems to, the currents carry larvae up and then between kind of Cuba and Florida. And that's ultimately what is probably settling in Florida over generations. I don't know if it gets there in 5 days with the larvae. But so those are genetically linked populations. So that is why we were given the permission from the Florida Fish and Wildlife Conservation Commission to breed the Florida elkhorn corals with the Honduran elkhorn corals. And then also Andrew was given permits to plant some of those back out onto the reef in Florida. So we have like 240 surviving recruits from those crosses. We only put 35 back out. That was just 2 weeks ago now. It was like in the very beginning of July. And they're just they're not even a year old. They're like 11 months old. So and we cut them in half before they went to save, that genet. So they were just little chips of coral, but they did go out onto a reef with Florida by Florida crosses as controls when we matched up the egg donors. So the Florida by Florida crosses are the similar parents to what were used to make the hunter. So it's not perfect. It's really hard to do this level of science perfectly with having to spawn the corals, make the crosses, settle everything, have kids, and have enough replication of every single combination to do the perfect study. Is like, scientists come to us all the time and they're like, so could you just cross like these 8 corals with those 8 corals and then make sure you get like 100 living babies from every combination? And that would be like, probably not.
I'll do it by Tuesday, no problem.
Yeah.
I lately find myself going, what do you think I am, Instacart? We're not, you can't just put in an order for like 100 of every specific genetic cross. But we find ourselves trying to do these things.
Keri, do you, I really like the little parrotfish protection 3D printed frames you put on those.
Yeah,
so
that's cool.
We've actually, inspired by that work, we've made a giant version to go over the reef stars that the Mars make.
Oh, nice!
And we're going to be trialing those.
Umbrella.
Yeah, we're going to be trialing those in the Maldives.
That's awesome.
Very cool.
Because Maldives has a huge parrotfish population, and it's controlling parrotfish grazing on juvenile corals is probably one of the biggest things in terms of outplanting.
100%.
Yeah, it is a massive issue.
And it's so site specific here. I mean, there is no feeling in the world like raising these coral babies and have them all go down the gut of a parrotfish within the first week.
Yeah.
And you're like, oops. and then you have to come back and start thinking of all these gadgets, right, that have to go on top of them. And we've done so many gadgets. I mean, we have bamboo skewers in a tripod, and then we're like, that kind of works. So then we 3D printed this little thing that was at least one piece that you could put over it. And then we made those out of biodegradable material, right? So we didn't have to come back and remove them. But then turns out they don't biodegrade nearly as fast as we thought they would. So it's like the parrotfish thing is a problem. And here, Miami-Dade is the worst when it comes to the predation. I don't know why. And some places in the Keys are bad, others are not as bad. And those umbrellas are made by a company called Reef Cells. They also make the plugs that we use that have a nice chunky thread on them, so you don't have to spin them a bunch. You just spin them like a couple times and they've locked in. So we have like transport racks that you just like put the plugs in. You can stack the transport racks in the coolers. And then those transport racks actually can fit even on a tree in the nursery. And then those can screw into receivers on the reef, and then the little umbrella screws in. So there's this whole system now.
And it's essential, right? Having that system connecting land-based facilities with in situ, transplantation is fundamentally. It's all about productivity and streamlining methodology. It's so important, the design aspects of this.
So I think go ahead, Chris.
No, you, Salem. It's all you.
Okay.
It's all you.
So I think as a final thing, I think this has been pretty insightful. We definitely need more time with both.
Yes.
This needs to be like a 6 hour, 5 hour.
I just realized it's over. I was like,
we didn't even get done. As soon as we get started. So that's the downside of this. But everyone has seemed to really like this conversation. So we need to have you both back for a long form one.
We'll do it.
So awesome. I think it seems like there's a lot of hope here. It seems like with the work of maybe manipulating symbiotes when they're young, durastinium, with the hybridization, there's cryopreservation, I believe you both have worked with. So, I mean, where do you think coral spawning goes next? Think like full automation's happening? What's the future look like?
Quick.
you have 3 minutes.
There's a project we're working on. If we get funding, you'll see some really insane stuff in a year or two. We're going stem cells. Let's go down that road.
Nice.
Wow.
Nice.
Very interesting.
I can comment too on the automation thing. I'm mixed on automation. There are some processes that 100% can be helped with automation. But honestly, there is still such a factor in this of just being a good aquarist and using your eyes, and you're seeing these animals every day in the tank and you're noticing that little thing. That's really, really hard to train into an AI system. I mean, people are trying. But there is such a human element to what all of us do in taking care of reef aquariums that it's going to be very, very hard to get to the point that all of this is automated. There's certainly some processes, like just if you could put a baby coral plug in a machine and it just cleans everything, awesome. But even that, you can damage it, and it's a balance.
Well, I would add to that as well. I mean, from my perspective of working with Mars and seeing how that methodology works, and it's fundamentally driven with the community involved. Mm-hmm. So I just say, literally this last week, 600 reef stars have been put down. It was a women's only program because of Women in Science this past week. 600 reef stars. That's 9,000 fragments of corals. It's asexual. But that will have included 100 community members engaging with that program to restore their own reef. And that is incredibly powerful as well. And how we can combine those two worlds, then great things can happen, I think.
You guys,
conversations like this are definitely a step in the right direction to getting things. Yeah.
And thank you for what you both do. I mean, what you both do is something that has been fascinating. I've been fascinated with for a long time, and being able to be a part of it has been just over the moon about it. I mean, and thank you so much for all that you've done and all that I've learned from both of you. And let's keep doing this.
Love you, Keri.
I love you.
But Thank you so much for all you've done and all the information you put out there. And thanks for not just hoarding it all to yourselves, because it needs to be out there for everybody.
Thanks for doing this. This is what it's all about as well. This is how communication with the world takes place. So we really appreciate being invited on.
Yeah, thank you guys so much. I was looking at the lineup, and you have a really, really unique group of people on this lineup, and I was actually really interested in. I'm like, I can learn from some of this. It's just awesome. Like, you've really kind of put a lot of effort and thought into making this 24-hour period just really diverse, having a lot of different perspectives, and I just really appreciate being part of it. So thank you.
Thank you.
Comment below if you want a 12-hour summit with Keri and Jamie sometime in the fall of the intermediate.
That would be fun. And we probably want to go longer after that, I'm sure.
Thank you, guys, and good luck with everything. Keep those monsters going.
Yep. More caffeine.
Yep. All right.
Thank you for coming on.
Take care. Cheers.
Bye, guys.
Take care, everyone. Bye.
All right.